This week, we'll be looking at the last group of diseases. Since we covered malaria last week, you will notice that all the vector-borne diseases today are actually viral diseases. By the end of this lecture, we should be able to describe the following human diseases: West Nile virus infection, Eastern equine encephalitis, St. Louis encephalitis, based on their clinical symptoms, epidemiology and transmission routes, laboratory diagnosis, treatment, and control and prevention. We will also try to describe the following vector-borne diseases briefly with respect to clinical symptoms, epidemiology, transmission routes, etiology, and prevention and immunization strategies if time permits. These diseases are yellow fever, dengue fever, chikungunya, Zika. Otherwise, the notes in the handouts are for reference purposes only, and you will not be assessed on this material that is not presented in the lecture. Arboviruses are a fairly large group of viruses that are arthropod-borne viruses. They are transmitted to humans by infected blood-sucking insects such as mosquitoes, ticks, and sandflies. The first arboviral disease we'll focus on is West Nile virus infection, due to the emerging nature of the disease in the past 20 years. West Nile virus infections have a wide range of incubation periods, lasting on average for a week. Symptoms are also variable from case to case. However, most cases, more than 3/4, are asymptomatic or may have flu-like symptoms, sometimes a mild rash and solid lymph nodes. This type of West Nile virus infection is now called West Nile virus non-neurological syndrome. A small number of cases, less than 1%, however, may become severely ill with neurological involvement and symptoms such as muscle weakness and/or polio-like acute flaccid paralysis, encephalitis, which is inflammation of the brain, meningitis, inflammation of the lining of the brain or spinal cord, peripheral nerve fiber degeneration may present clinically as well. These cases may become life-threatening. This type of West Nile virus infection is called West Nile virus neurological syndrome. Some severely symptomatic cases recover completely. However, others suffer long-term effects which may be physical, such as long-term muscle weakness and paralysis, fatigue and headaches. Cognitive problems with confusion, depression, problems with concentration, and memory loss may also be present, as well as functional disabilities with difficulty preparing meals or going out or shopping. West Nile virus infections that lead to serious health effects are more commonly found in certain higher risk groups, such as cases with increasing age or weakened immunity due to chemotherapy or chronic diseases such as diabetes, cancer, heart disease, and alcoholism. West Nile virus infections are transmitted by mosquitoes, mainly cholocynes, that are fed on the blood of infected birds, mainly those in the corvid family, such as crows. Mammals, including humans and horses, are dead-end hosts because viral levels are too low to be transmitted to another host. There is ongoing surveillance of West Nile virus infections in birds, and every year specimens are collected and submitted for testing. You can see from this table that Ontario and Quebec are the two provinces that send in the most specimens for testing and also generally have the highest rates of infected birds. Even though in 2018, Manitoba had a very high percentage, more than 3/4 of the birds submitted were positive. I've added the data from 2012 as well, because there was a peak number of human cases that year in Ontario and Toronto, and I thought it would be a good idea to compare bird infection rates with human ones. In terms of its history, West Nile virus was first isolated in Uganda in 1937. It's really a tropical pathogen that didn't have any impact on North America till just over 20 years ago in 1999, when the first recorded outbreak was reported in New York City. In 2001, the virus was detected for the first time in Canada in dead birds and mosquito pools. A year later, infections in horses were reported, as well as the first suspect case in a human. No one really knows how the virus made its way to North America. From molecular epidemiological studies, the genetic type of the virus suggests that it originated from the Middle East. So, it is possible that an infected migratory or imported bird may have brought it across the Atlantic, or an infected mosquito that was transported to North America by accident on a plane. By 2002, there were more than 4,000 cases of West Nile virus infections in North America. In the past 10 years, we've seen incidence rates peak and then fall back to baseline levels. National, provincial, and municipal rates show the same trend, with a sharp increase in cases in 2012, which is especially marked in Toronto. I will provide you with more information on the next slide. From the previous slide with the graph, we saw that the rate in Ontario and Toronto followed the national trend. However, the incidence rates in Ontario and Toronto are slightly higher than the national rates, and depending on the year, more than half the Canadian cases were reported in Ontario residents. In fact, Ontario and Quebec have the highest number of cases in Canada. We will see that in the next few slides. Toronto actually had a slightly higher incidence rate than the rest of Ontario, and there have been nearly 100 cases per year during peak years such as 2012, during which the highest incidence rate was observed since West Nile virus infection became a reportable disease in 2003. If we look at the very last Toronto public health investigation in 2016, the top exposure setting was in a private home. All 22 cases in 2016 acquired their West Nile virus infections in a private home, and only less than 10% traveled within Ontario. This means that all cases were actually in their own homes when they were bitten by an infected mosquito. If we look at the 2012 investigation, where there were more cases, you can see that the results support the observations in 2016. We can see that the infections in the majority of the cases were acquired locally, although in this study we don't have details on whether this was in their own private home or outside in a park or other recreational setting. Just over a quarter traveled within Ontario, and a minority, less than 10%, traveled abroad. Transmission of West Nile virus is seasonal due to the higher mosquito vector numbers in the summer. We can see from this figure that the number of cases in 2019 was lower than the average reported in the previous 5 years, especially in mid-July. The dotted red line represents the cases that were reported in 2019, whereas the black line, the mean from 2014 to 2019. This seasonal trend of a late summer peak is also observed in other earlier years, and even though there was a peak in 2012, you can see that this peak is more of a blip when the numbers in 2007, which is the blue line, are included in the graph. Since we only look at the past 10 years, we totally missed this massive 2007 increase in case numbers, and that is why I've included this graph, even though the data is older. If we look at the numbers and the geographical locations of cases, we can see that both Quebec and Ontario consistently have the highest number of cases, and for the first time in 2018, rates per 100,000 population were provided, and these are included in brackets. The rate in Quebec in 2018 was twice as high as in Ontario, where the rate was slightly lower than the national rate. When we look at the different types of West Nile virus infections reported, we can see that more than half of the cases had the non-neurological or milder form of the disease, and just under a third had the most serious neurological form. When we look at the Canadian map and the geographical distribution of cases, we can see that 2019 was not a very active year for West Nile virus transmission. Areas with a handful of cases included southern Alberta, the Montreal area in Quebec, and eastern Ontario, as well as the Golden Horseshoe area, including Toronto. However, since these are raw case numbers rather than rates, there is a bias towards more densely populated areas. West Nile virus is a flavivirus and is closely related to other viruses that cause yellow fever, dengue fever, and St. Louis encephalitis. It is a single-stranded positive sense RNA virus with an envelope that contains hemagglutinin and lipoproteins. You can see the transmission electron microscopy picture of this virus on the right, which has been color enhanced. Lab diagnosis of West Nile virus infections are determined by serological tests that detect West Nile virus specific IgM antibodies in serum or cerebrospinal fluid. There can be false positives due to cross-reactive antibodies after infections with other flaviviruses. All positive results obtained need to be confirmed by antibody neutralization testing of acute and convalescent phase serum specimens using hemagglutinin inhibition or plaque reduction neutralization tests. If you've never heard of these tests, I'll describe them now, as they're rather complex and confusing, so it may take some explaining. Normally, to look for the actual pathogen or the toxins it produces, we can do a cytotoxicity test by adding the pathogen or its culture supernatant to a cell line cultured in vitro. This is the first test above. If the cells are damaged, we can confirm that the toxin is present and the case is infected. However, in antibody neutralization tests, you really want to see the opposite because you're trying to detect the presence of antibodies that will neutralize the toxin or pathogen and prevent cytotoxicity from occurring. So if we look at the second test below, you can see that toxins are mixed with the antibodies, which will be the test serum from the probable case and the cell line. If there is no cell damage, it means that neutralizing antibodies are present and the case is lab confirmed. If cell damage is observed, no neutralizing antibodies are present, and the case cannot be confirmed. The same principle can be applied to hemagglutination tests and hemagglutination inhibition tests, which are used in the lab to diagnose viral infections. In the first test above, when a pathogenic virus is mixed with red blood cells in the absence of antibodies, hemagglutination occurs. However, in the second test below, when serum from a case infected with West Nile virus is added to the test, and if the case has neutralizing antibodies, as in the test below, no hemagglutination occurs as it is inhibited by the antibodies and the case is confirmed as positive. So just to go over the hemagglutination inhibition test again, this time using some test wells to make it more realistic. In the top row of the picture on the right, you can see several wells with test serum added to the red blood cells and West Nile virus hemagglutinins. The picture in the middle is the real-life picture of the fourth and fifth wells of the test strip in the top row. Agglutination is seen in the fourth well, indicating that the serum specimen from this case does not have any neutralizing antibodies to the viral hemagglutinin, and therefore the case does not have West Nile virus infection. This is a negative result. In the fifth well, we see the non-agglutinated red blood cells that have sunk to the bottom of the test well. Since there's no agglutination, the test result is positive, as neutralizing antibodies are present that are able to inhibit the agglutination reaction. This case is therefore confirmed by the lab test to be infected with West Nile virus. There is no specific treatment, medication, or cure for West Nile virus infections, and supportive therapies such as analgesics can be provided to ease symptoms and prevent secondary infections. Since there are also no vaccines available right now, the best prevention strategy is to avoid mosquito bites by using insect repellents and wearing protective clothing such as long sleeve shirts, long pants, and hats which are light colored as mosquitoes tend to be attracted to dark colors. Integrated pest management can also be used to control mosquitoes by exclusion with tight-fitting intact doors and window screens. Since mosquitoes lay their eggs in standing water, and small amounts of water can act as breeding grounds, mosquito breeding sites should be eliminated around properties by draining standing water regularly, twice a week, and removing old unused items that tend to collect water and changing the water in wading pools, bird baths, pet bowls, and livestock watering tanks. Also, eliminate mosquito breeding sites around properties by purchasing an aerator for ornamental ponds to keep the surface water moving, making it inhospitable to mosquito larvae, covering rain barrels with screens, and clearing out eaves troughs regularly to prevent clogs that can trap water.